Yes, thank you very much for the invitation. It's a pleasure to be here. I'll try to give you an overview of what we've been doing in the last couple of years in understanding the dynamics of biomolecules with simulation and machine learning. We also use we don't do experiments, we use experimental data, but today I will not talk about the work with experiments. So I think you all know that we do molecular dynamics. Many people, us included, do molecular dynamics simulation of proteins or like biomolecules in general. What is a molecular simulation? We have a protein or a macromolecule represented in atomistic details, and we integrate Newton's equation of motions, classically, usually, to observe the dynamics of this molecule. But there are three, I would say, challenges in doing this we all face. The first one is the force, what is called the force field, that is the Hamiltonian, the energy function. Because in principle, if you want to be really first principle, one would have to write the Schrödinger equation for a system of atoms. These are atoms, many atoms, especially if you put the protein in water, there is a lot of water molecules. And if you want to be a first principle or obtain a first principle description, you need to write the Schrödinger equation. But as you know, if you go beyond the hydrogen atom, you cannot do much more exactly with the Schrödinger equation. So invoking the separation of time scale between the electrons and the nuclei, people say that the quantum effects are mostly in the electrons, and then they produce an effective energy function for the nuclei that you can use to move them classically. But nobody knows what is this effective Hamiltonian, what is effective energy function to move the nuclei. There has been a lot of work in the past 75 years to define empirically these functions, and they work okay. They work actually quite well now after 75 years of a lot of work from many people with some quantum mechanics, with some experimental data. Now machine learning is changing that, and people use a lot of machine learning to define the energy functions. But even if you have the energy functions, how do you integrate in a computer such a large system in a way that you can sample events that are very long in time scale for biological system, meaning you want to observe a molecule perform a biological function, and this usually happens in milliseconds and beyond. But in order to consider the fastest time scale to integrate this equation, that is femtosecond, is the vibration of bonds, and then from femtosecond to millisecond, there are too many order of magnitudes to do all these steps to sample them in a relatively fast time. So can we, even if we have the energy function, given we don't know if we have it, but assuming we have the energy function, can we simulate it long enough? And oops, okay. Even if we simulate long enough, what do we do with all the data? We get a lot of data, a lot of coordinates as a function of time series with millions of coordinates for millions of time steps. How do we understand what's going on there? And machine learning is making a lot of progress in force field development, in time scale, in speeding up the simulations, and also in the analysis. I've worked in all these fields, but today I mostly talk about what we are doing for the time scale, a little bit for the force field. I will not talk about the analysis today. So before I start, I want to address the elephant in the room. When we talk about machine learning, of course, in biology, everybody thinks about AlphaFold, Nobel Prize in Chemistry 2024, and the big splash in the news that now you can predict structures from sequences in proteins. And that's great. That's a wonderful tool. We use it too. What we want to do is slightly different. We want to, if AlphaFold gives you one structure or a set of structure, you can think about these as points in a varidimensional landscape. These are a set of coordinates, and there are points in a dimensional surface. What we want is the full surface. We want the full, what we call energy landscape. We want to understand not just some interesting point like the deepest minima in this landscape. We want to understand all the minima. We want to understand the transition state, the subtle points, how the molecules in general moves on this landscape, because we believe that dynamics is an important factor in the function itself. So just looking at static structures doesn't tell the whole story. We would like to put dynamics into the game. Of course, this is easy to say, but as I mentioned at the beginning, if we consider the time step for molecular dynamic simulation, we need to follow the fastest process, that is the vibrational bond, that is femtoseconds, and we want to go to observe processes milliseconds, second, even beyond. Then realistically, even with the fastest computer, you cannot go much beyond microseconds. You can do to milliseconds with special purpose computers that only few in the world exist, and they are fully dedicated to that. We want to make it in a different way. But let's look at this from a different point of view. I come from statistical mechanics, and in statistical mechanics, as you know, there is this usual description of microscopic and macroscopic and how to bridge them. What does it mean? Microscopic is one small molecule, like one water molecule. And in this case, you know what you want. You want to find the electronic structure, you want to find the molecular orbitals that tells you the whole story of this molecule, or how these molecules interact with the world. But if you have an infinite number of water molecules, you have bulk water, then it's a different story. You are interested in a different set of variables. You are interested in collective processes, what we call emergent properties, things that exist because you have many, many components, not just one, like thermodynamics, temperature, pressure, and so on. The phase diagram is the important point. So if you change the number of degrees of freedom, even if it's the same substance, water, you change the point of view. You go from microscopic to macroscopic. And what we are interested in biophysics usually is in between. It's not as microscopic as one water molecule, it's large molecules, but they're not in the thermodynamic linear. They're not an infinite number of atoms. And also they're very heterogeneous. They have interactions with discontinuity, interaction interfaces, interaction solute solvents, large clusters, and so on. So what is the point of view we need to take to describe this system? They are neither microscopic nor microscopic. They are in between. So I think it's relevant to ask, what are the variables really that characterize this system? Do we really need all the atoms, the position of all the atoms to understand what's going on? And what is the resolution we need to use for this system? Because they are because they are not really microscopic, we don't need the most accurate resolution. We don't need to take a first principle description. And so one big effort in my group has been to design models that are minimalistic, meaning they're as simple as possible, but they still preserve the relevant physics. They are quantitative and minimalist. And I would argue that this is not just speed up simulation. Of course, we do it. The practical side effect is that we can speed up simulation quite significantly, but also we can understand what's important and what's not important when you go in the direction from microscopic to macroscopic. So it's a more general point of view. And I would hope to convince you that with the results I'm going to tell you today, I think we have good evidence this is possible. Of course, we are not there yet, but we have good indication that this is a pursuit that is worth pursuing. So this is coarse graining. So we go from here, this V of R is, let's say, first principle potential, something you have, in principle, you can have a machine learned potential, something as accurate as you can be to describe the energy of interaction of the different atoms. Then you start, so this would be the representation of a protein, a small protein in water with all the atoms. Then you say, okay, I want to do the same thing. I want to reproduce the same dynamics, but I don't want to move the water around because it's just, maybe it's just a field. I can just introduce a field to reproduce the effect of the water. But then I can group the atoms together and I can have some sort of beads to represent group atom. And what's the size of the bead? I can get coarser and coarser and coarser. But as I do that, while if I am from first principle, I know that there are electrostatic interactions, there are dispersion forces, there are bonds and so on. Here, I don't have this description because I group things together. So how do I describe the interactions between group of things? So as I increase the coarsening, I lose the ability to assign an energy function a priori. I lose the form of the energy function. And this is the biggest problem in coarse graining, that you don't know what the energy function is. But there are different strategies to try to find an energy function. You can go top down, meaning you say, okay, I don't care about what's inside. I just want to reproduce some set of experimental data. And if it works, it works. This is the strategy taken by Martini mostly. That is one popular coarse grain force field. And it used, they use parameterization, they parameterize this force field using transfer free energies from different environments. And then they use it for simulating proteins, membranes, and so on. It works very well for membranes. For proteins, no, it's not so quantitative. Then you can say, I don't care about reproducing experimental data. I want to do some sort of, I want to visit certain structures. I want to be in certain structures as the energy minimum. And this is other force fields are designed, other coarse grain force fields are designed with this idea. Or you can say, I want to go bottom up. I want to really start from the fundamental description, the first principle description, and mathematically integrate out degrees of freedom, renormalize, as we do in physics. The formalism is well known. And this approach has been taken by different people. Most popular is the Greg Voss and Wilnoid MSCG multi-scale coarse graining approach. So this has been well known for a while, and we decided to follow the bottom up. We also do some top down and we combine them, but today I'll talk about the bottom up mostly. Let me explain a little bit how the bottom up works. So you want a potential. You start with a potential V of R. This is the atomistic potential. This is the first principle potential. And you want a coarse grain potential such that the thermodynamics is the same. The thermodynamics of the microscopic needs to be the same as the thermodynamics of the coarse grain. What does it mean? It means that the free energy needs to be the same, means that the probability distribution needs to be the same. You want to match the probability distribution. So this you can write mathematically. The probability distribution of the coarse grain potential is the marginal probability of the Boltzmann distribution of the atomistic potential. Assuming you are in the Boltzmann ensemble, for these two to match, this relationship needs to hold. And because we have a probability, we take the log, we have an energy. And so with this, assuming we are in the Boltzmann statistics, these equations need to hold to have thermodynamic consistency. If we define a coarse grain potential according to this equation, the thermodynamics is the same. So we can use the coarse grain potential, you obtain the same thermodynamics as in the atomistic potential. much faster. Okay, so this is, in principle, you have a definition, but in practice, this integral here is very nasty. You have a delta function inside that mixes everything together, or the degrees of freedom together. So even if you have two bodies only interaction in this first principle potential, in this integral, everything gets mixed up and you get the emergence of multi-body functions. And this, you don't know what you get because you cannot solve this integral. Imagine this integral is, you're integrating millions of coordinates. So you cannot do this integral, not even numerical. This is an explanation, this is a representation going from 2 dimensions. You have this 2-dimensional potential. You want to obtain the equivalent thermodynamic potential in 1D. So you integrate out the y dimension. Here you can do it because it's just in a 1-dimensional integral. You get this potential here. And if you do the thermodynamics here, for instance, the relative free energy between the 2 minima and so on, or the height of the transition state, you get the same here and here is equivalent because they're thermodynamically consistent. So we want to do the same, but with millions of dimensions. This is not possible directly, but people have worked out, starting from the 90s, from Ercolesi and Adams in 94, that this thermodynamic consistency can be reformulated variationally. You can have a variational principle that says that if you define a function as a function of this potential u, and you let the potential, this trial potential, span the whole functional space of integrable functions, then the absolute minimum of this functional that is defined here is exactly the one that satisfies the thermodynamic consistency. So if you define this functional, and let's look at this functional, there is here u is the potential you want, is this the coarse grain potential. You have a gradient, so this is a force. And here, this F is the atomistic force, the force of the microscopic model that is projected with this operator on the coarse grain space. This is the atomistic coordinate that is projected with this operator in the coarse grain space. These are parameters. So you need, this is called force matching because you need to match the forces. Not really much because this is a projection. So it's called force matching, maybe it's a bit of a misnomer, but you need to minimize the fluctuation of the forces. And these operators here are projections. Let's say you can consider a projection if you, instead of all the atoms, you can pick some of the atoms. That is slicing projection. You pick some representative atoms. And this is the equivalent in the force space. So if you let the potential u, the trial potential u, span and be any possible function, then the absolute minimum of this functional is the same as solving this equation. So this is really useful because you can solve things variationally. The problem is if you pick a functional form, let's say, I use exactly the same functional form as an atomistic force field, you restrict this functional space too much, and you may be very far from the real solution. So we had the idea this is the perfect case for machine learning because we always say that neural network are universal function approximator. Well, let's take a neural network here, and then we learn from data. We don't need to impose any functional form to you. We learn it. And this is what we did. We started in 2019, and fast forward 5 years. This is what we can do now. So we've used this idea, and we generate a lot of forces from atomistic models. And then with this loss function here, we take a data-driven version of this force function. This is an integral, but we generate it from data. We minimize this functional, and U is a neural network. And we learn this neural network with the parameters of this neural network. This is what we can do. This is the all atom reference. This is the free energy. So this is for this small protein here. We can do it I'll show you later for a much larger protein, but to do a comparison with all atom, we need to use a small protein because otherwise we don't have the comparison, we don't have the reference because it's too expensive. So this is the Olatom reference. These are simulations. These are months and months of Olatom simulation, even if the protein is small. And then we project, we compute the free energy, we project it as a function of two collective coordinates. We find that there are three minima. This is the free energy scale. So there is a deepest minimum that corresponds to the folded state. Another minimum that corresponds to the disordered unfolded state. And another shallow minimum that corresponds to the misfolded state. If we do the simulation with our model, with our CG model, it takes less than 1 day, and we get a very comparable free energy. Same three minima. I would say this is quantitative. It's within 1 kilocalorie per mole from the reference. How do we get there? So there is a lot of technical development I'm not going to tell you about, but one of the latest things we've done that are very important is to use this force matching, this functional that we use as a loss in combination with denoising diffusion. This is in archive. If you're interested, we can talk more about this, but I just want to give you the idea we are using here. So this is the force matching loss that written a different way, what I written in the previous slide. So this F is the atomistic force, the label that we get from the atomistic simulation. This T is the projection. And this is what we learn. This is our neural network. This is the force of corresponding to our model. This is the gradient of our model. So if we put here a gradient of an energy and we learn the neural network is the energy, we learn the energy. What do they do in the denoising score matching loss? This is what is at the base of a very popular diffusion model. Now everybody uses diffusion model in machine learning. For instance, AlphaFold 3 is a diffusion model. There are generative models that learn to denoise. What does it mean? This is the loss of a denoising diffusion model. You define a force that is a noising force. So you have your coordinate are R. You apply a noise kappa that moves the coordinate just a bit according to certain conditional probability. For instance, can be a Gaussian conditional probability distribution. You start from R, you noise it to R prime. Then you take the log of this probability distribution, then you take it the gradient. That becomes a force because the log of a probability distribution is an energy. The gradient of an energy is a force. So you're defining a noising force. And then you put a neural network here to learn the noising force. And then if you learn the noising force, you can remove it from the data and you learn to denoise it. And this is how the diffusion models work. But there are a lot of similarities between these two. In both cases, you learn a force. So we said, why don't we combine the two things? So you have your coordinates, you noise them a little bit, then you coarse grain them, and then you have a coarse graining force, the label force coming from their simulations, and you have the noising force because you have noised the data. And you sum them up. How do you sum them up? With you can combine them. You can combine it with some hyperparameter, but we combine it in order to minimize the noise. No, these are technical details. But anyway, you can combine the two things. And this works very well. Why does it work well? Because here you have information about the original Hamiltonian. The force is a gradient of an Hamiltonian, of an energy function. Here you have distributional information. You learn from the probability distribution. So these are completely different information. You combine them and you get much more powerful method. So just to show you how it works, this is the larger protein until 9. This is the unfolded state. This is the folded state. We want to learn an energy function that can fold and unfold this protein as you could do in an atomistic simulation. So we cannot do atomistic simulation of this protein, it's too large. So we take what has been done on one of these special purpose computers in the show research. And if you can do it in equilibrium, you get this, as a function of one reaction coordinate, you get this gray distribution. And then if you use either diffusion models or atomistic forces, you can capture something very similar. But this has a lot of data. If you use just 2% of the data, if you use either the denoising by itself or the atomistic by itself, you cannot fold it. You see this red curve is only on the unfolded state. This is the folded state. The protein stays disordered. If you combine the two things, you recover the free energy very well. So we have introduced a lot of technical development that allows us to push this idea quite far. But the ultimate goal is to develop these in a general form. We want to be transferable, meaning we want to develop a force field that can be applied to any protein, not to learn one protein and then apply to similar proteins. We want to learn a small protein and then do big things that cannot be done with all atoms. So this is a very ambitious project that has many different components. I'm going to tell you very little about the different components, but to give you an idea, and then I want to show you some of the recent results. So one of the most important aspects of this is the data set, what to learn from. And we spent a lot of time creating the data set. These are, remember, the labels are atomistic forces. So we need to learn simulation, and we need to do it for a diverse set of structure and sequences, and also small peptides and monomers and dimers coming together at different distances and so on. This mapping, this projection, these are important. These are more technical details. This is a bit of StatMech here to optimize this projection. What we call the priors. So if we know, we have some information about the physics of this, is a polymer, first of all. Protein is a polymer. There are some bonds. There are some forbidden geometry. You cannot put two atoms on top of each other. Each atom has some sort of excluded volume. So this is all information that you can put into the model. It's what we call the priors. The graph neural network, we have done a lot of work in designing the graph neural network that is consistent with the physics. And I'll tell you something later on our latest development on the graph neural network itself. And then the validation. So as I told you, we have a very large data set of proteins. The mapping is such that we optimize how we combine the atoms. The priors use, we use bond angles, dihedrals, and excluded volume. The graph neural network mostly we built from SNET, that is a popular neural network. We also use Spain, Maze, all the popular neural network, and then we develop our own addition to it. We need to make compatible with our problem. And then the simulation, then we do molecular dynamics, Langevin dynamics of the resulting force field. So this is our preprint. We just submitted the revised version of this paper. I'm going to show you some results on this transferable coarse-grained model. So this is the data set, and even if it's large, it's actually quite small for machine learning standard. It's 50 proteins that are not larger than 75 residue each. They're mostly around 50, but we picked them to be as diverse as possible in terms of sequence and secondary structure composition. And then we have a lot of fragments, dimers, at different distance from each other. The projection already mentioned, the priors already mentioned, and the neural network is mostly degenerate. Let me show you the validation. These are proteins that were not in the data set. So these are extrapolation. These are a prediction of the model. They have very little sequence overlap with the data set. So we can fold all the proteins for which we have the folded landscape, for which we can afford atomistic simulation as a comparison. We can fold and unfold all of them quite well. You can see the overlap with the folded structure in the PDB. We can analyze the folding landscape. This is our model in comparison with the existing coarse grain model. For instance, this is Martini. Martini cannot fold the protein. You see, you don't get folded state because it's not designed for that. But even other models cannot fold proteins, or if they fold them, they only can fold it. They cannot explore folding and folding and so on, because they were designed for different purposes. Our model is designed to match the thermodynamics. We can also explore the landscape of proteins that we cannot do atomistic simulation of. So for instance, this is S6. There are no atomistic simulation folding of this protein, but with our model, we can do it. We recover the folded structure, and also we get some disordered structure as unfolded state. We can do larger protein, and we can compare with very stable proteins like this one. This is Q is the fraction of native contact. RMST is the root mean square deviation from the native state. So this is the native state. You see, this protein is very stable, mostly populate the folded state. We can do simulations with all atom all in the folded state that compare the fluctuations, and we get a good qualitative agreement. I want to show you more about this protein. So we start from the completely extended protein. We take just extended polymer, and we let it go. And you see that quickly find the folded state, and then oscillates around the folded state. So mostly you find oscillation around this state, that is the folded state, but also sometimes you go here. This is the mirror image of this. Two helices are swapped. And interestingly, this protein was designed starting from a helix bundle, and during the design, they found this as well. And then they tried to design it out, but this was observed. So our model recovers also this alternative structure. We can do intrinsically disordered proteins because we are not limited by the time scale. So we can explore the full energy landscape of an intrinsically disordered protein. These are proteins for which there is no stable, folded, well-ordered state. The protein is very flexible and can explore different states. For this protein, there are a lot of, there are 50 NMR structures. So this is our landscape as a function of the two collective coordinates. We find that all these gray crosses are the NMR structure, are all collapsing into the deepest minimum in our landscape. So the deepest, the most stable minimum, it corresponds to an NMR structure. But we also observe other states. And these states are very metastable. You see, there is there are several kcal per mole difference between this state here. This is the scale. So there are at least 4 or 5 kilocalories per mole between these states and this state. So either maybe it's not found experimentally because it's not very stable, or another thing we tried, we took structures from these alternative states. We reconstructed in all atom details, and we started all atom simulation from there, and they stay there. They don't move. So atomistically are stable. So maybe if they're not real experimentally, is what they inherited from the atomistic force field, but you never see them in atomistic simulation, because if you start from here, it will take forever to go here. Then another thing we are very happy about, is mutational analysis. So in our model, because we can do folding and folding, we can do it also for different mutants. And then we can compare the difference in free energy upon mutation. We can compare with experimental data. And this is the agreement. So we get an agreement that is comparable to atomistic simulations. We get a pretty good correlation, and 1.25 kilocalories per mole from SPEC. So this is becoming quite, as I said, is the accuracy of atomistic simulation. So we are very happy, and we're collaborating with the experimentalists to refine. We can do some top-down to refine this value tweak the parameters to be in better agreement, although experimentalists too have an error bar. So usually you cannot trust experiments more than 1 kilocalorie per mole. Also, we can do folding upon binding. So this peptide is called PUMA, is intrinsically disorder, but if you put it in close to its corresponding protein, there is an induced fit. The peptide folds and binds in 28. So we decided to try it. We took this protein. The peptide in isolation, this is the RMSD from the helix. The peptide in isolation is this blue curve. It's all over the place. It's an intrinsic disorder. But if you put it next to its own protein, this is what you see. You see the RMSD collapses very quickly because very quickly the peptide folds and binds. And then we said, okay, maybe it does it with any protein. So let's put it next to another protein, ubiquitin. It doesn't fold. It goes around and doesn't fold. So there is we could learn the recognition of the peptide to the protein. These were not in the data set. So this was the model learned something about the interaction of the different amino acids to make this prediction. More recently, we started to also work on membranes because a lot of proteins are working in membranes or in membranes. So we extended our model to membranes. And now we have a stable membrane model. We can also see the formation of membranes, micelles and stuff like this. And there is no water. This is just the water is a field as for the protein. Now we need to take the step of making the proteins in them. And another thing we are really interested in is the interpretation of all of this. Because we learn a force field, but the force field is a neural net. What does it mean? We don't see it in the terms of sum of two bonds and so on. So we would like to understand what's going on. So we do together with Klaus Robert Müller, who's done a lot of work in Berlin in the interpretation of neural network. We did something that is basically a tailored decomposition. Machine learning, they call it layer-wise relevance propagation, but in physics terms, it's a tailored decomposition or sensitivity analysis, basically. And we can extract all the two-body, three-bodies, and so on interactions from the neural net. First, we did it on liquids to see how it works. So we took methane, we coarse-grained methane from its atomistic form to the central atom. And this is we did it with two different neural network architectures, Pain and SO3net. They are very different architectures. They're both equivariant, but they're different. And if you extract the two-body interactions, it's basically the same. It's just a repulsive wall. If we do the same thing with water, with coarse-grained water to the central oxygen, you find an attraction beside the repulsive wall. You find the deep attraction in the two-body interaction. That is the hydrogen bonding. Then we can look at the three-body. The methane doesn't have three-body. This is the distribution of the angles. The contribution of the three-body in the membrane is 0. That means that you don't need the neural network to coarse-grained membrane. You can use these interactions, two-body interaction, it works. And the methane is very easy, just hydrophobic repulsion. But water is much more complicated. There is an attraction corresponding to the angle that is the tetrahedral arrangement. So it learns the tetrahedral arrangement and everything. Then we can do it with proteins. This is for this protein here. This is the folding landscape with our model. And you see in the folded state, the two-body interactions that are relevant. Here, blue means attractive, red means repulsive. You find all the attractive interaction in the secondary structures. This is the contact map. So these black area are the secondary structure formation. So you see that all the contacts that are relevant are the ones forming the secondary structure. In the unfolded states, there are relevant contacts, attractive or repulsive everywhere. We can look at what are the most relevant two-body and three-body interactions, and you see that they're all in the core. These are the two-body, interesting, they are repulsive two-body interaction in the core, but everything else is attractive. These are the three-body, the most important three-body interaction. And then we can do mutation again. We can look, when we do the mutation, we can look at how the network changed. So if you mutate here a leucine into a phenylalanine, these are the contacts that change the most. There is a distant contact that is not in contact with this residue 30 that changes because this is through the network. And if you mutate isoleucine into aspartic acid, you disrupt this network of interactions connecting to isoleucine. And you can see it in the contact map that are attached. I mean, there are also interactions that are not directly bonded that are affected through the. Okay, the last thing, do I have another 5 minutes? Last thing I want to tell you is more on the neural network side. So we use graph neural network a lot because molecules are graphs. But one of the main limitations of graph neural networks is that they are short-sighted. What does it mean? They have a cutoff radius. This is a power of graph neural network. They're fast because they're local, but there's also a limitation because if you have a molecule, if you consider this site of the graph, this node of the graph, this node sees in the first layer a certain cutoff, and the second layer another cutoff, but it's not infinite, it's finite. And we know in physics there are interactions that cannot be truncated. They are not short range, they are long range, like electrostatic. 1/r cannot be truncated. If you truncate it, if you consider only the effect in a certain cutoff radius, the error is compounded, it becomes too large. So how do we take this into account? There are different solutions. One could be that just don't put a cutoff, go to infinity. Well, that goes like n squared. Not nice. Too expensive. If we want to scale up to very large system, you cannot afford n squared. Add a network in reciprocal space. That means beside the local neural network, you can put you can do a Fourier transform of the distances and put a graph neural network in Fourier space and then transform it back. This is a way to take into account, to go from the other end, to go from the infinite closer. And if you engineer the two things to match in the middle, you can capture it. Scales like n log n, and there is a large prefactor here. So we tried it. Kind of what I'll show you later, but it was too slow. You can, other people have just put 1/r manually inside the neural network. And say, okay, I know that I'm missing 1/r, but if you do that, and we cannot do it in our network because we have a Gaussian model, we have multi-body terms. How do we put over R multi-body? We cannot do that. So we decided, let's go with a different architecture. I put two bright people, a graduate student and a postdoc, and they had a lot of fun for many months, and now finally we have a working model. So this is the idea. We call it range. That is, I don't remember what is it. Something about relaying attention network. Anyway, they like the word range, so they made up an acronym. So the idea is the following. We have a local message passing. This is the graph neural network. We start from a point, you have a cutoff, you go through your layers. Then we introduce master nodes or global nodes. These are nodes that see everything. They see all the nodes in the network. All the nodes talk to the master nodes. The master nodes are located in the center of mass of the node. We can have many of them. All the nodes with the attention mechanism, communicate to the master node. Then the master node, all the different master nodes, broadcast information back to each single node. But this go from many to one and from one to many. So this is linear. It scales like the number of the length of the clothing. So the graph. So it's linear. But in this way, all of them go into the master, each master node. Each master node goes back to each node. Everything is mixed. So you can connect very long range information. And in fact, so we do different steps. We do the local message passing, the aggregation, the broadcasting, and then we go back to the local message passing, and we keep going, and we have a global message passing. We tested it on the Acquiem Aquamarine dataset. That is, this is we tested it to reproduce atomistic force field. So we tested to reproduce quantum mechanical data with atomistic energy functions. So this data set is built to look at long range effects. They used, this is from Alex Tachenko, who has developed a quantum mechanical approach to incorporate long range effects. And so these are quantum mechanical data as a reference. And as I said, these are very large systems, so you need to include electrostatic and long range effects. And these are our results. So if you use SNET, that is one of these global message passing, you cannot go, the errors decrease a bit as you increase the cutoff, but then it saturates. If you put our range global message passing, you go down dramatically. And if you use pain, that is another graph neural network, is more expressive than SNET. So you start lower, but with our range, you go much lower. And this is the cost. There is a fixed cost only on top of, so for different cutoffs, the cost is mostly the underlying local message passing. The global message passing is just a fixed step on top of the local. So it scales linearly. And if we compare with this network that had the Fourier transform, the graph neural network in Fourier space, they don't improve much beyond this is the max, this is the baseline. This is with the addition of this network in Fourier space. And this is, if you try many different models with many different cutoffs and so on, the lowest you can go is here, and we go much below. This is with SNET again, and this is with this is for the energy of the forces, the errors, and this is with pain. We go, and these errors are pretty small. This is 7.4 millielectron volt per Armstrong in the forces. These are very small. And we can do now with these are very stable. We can do molecular dynamics for many nanoseconds. For these are at the quantum mechanical detail. So this is, instead of doing ab initio molecular dynamics, we do with this force field. They're very stable. They don't explode. We can have this polymer folding, unfolding. It's not a protein. It's just a polymer, and it's very stable. And then we can also interpret, because we use attention. So we can look at the attention in aggregation. So what are the most important attention weights of the nodes going up to a master node? And then in broadcasting, what are the most important attention weight from the master nodes to the different nodes? And you see that it's really learning global information. For instance, here you have, this is for this molecule. that I showed the molecular dynamics of, you see all the hydrogens of this molecule communicate with this oxygen here. Or here you have that all the internal hydrogen communicate with the carbons, with all the carbons. So this is really distributing the information across the whole molecule. And with this, I would like to thank all the people who have done the work. I have a fantastic group, very hardworking people. I'm actually hiring, if you know PhD, postdoctoral candidates that would be interested, I'm really happy to talk to them. And my collaborators and all the funding agency and all of you for your attention. Thank you.